Hello, in this session we'll be touring the Ensembl database. Ensembl is a joint project of the Sanger Institute and the European Bioinformatics Institute, and is one of the three major web-based genome browsers. To get to Ensembl, open a browser and type in Ensembl, no E at the end, .org. And that takes me to the Ensembl database. It actually takes me, in my case, to the United States East mirror of the Ensembl database. And what you might notice on the front page is over on the left menu you see three popular genomes, human, mouse, and zebrafish. There are other genomes in Ensembl. In a pull-down menu that you can see here, you see below human, mouse, and zebrafish, some primates, rodents. You find that Ensembl itself is heavily focused on vertebrates, specifically chordates. There are other divisions of Ensembl that focus on protists and bacteria and other species, but the main Ensembl database is primarily eukaryotic. You can see that the current version in this video is release 73. Approximately every 2 to 3 months a new release comes out. And there is a search window that I can choose the species and do a search. So I'm going to jump right in and click human. And I'll go to the BRCA1 gene, BRCA1. And when I hit search, you'll see that the first hit says BRCA1 human gene. Below it you'll see BRCA1 human transcript. Well, let's click the gene for now. That will include all variations of the transcript. So I'm going to click BRCA1 human gene. And what you might notice is that this particular gene has 31 known transcripts or splice variants. That's exploded. So more and more splice variants are being found all the time. And you can see the transcript ID, ENST, in the transcript length, along with a protein ID, which is ENSP, followed by the protein length. So the first transcript is 7,094 nucleotides in the mRNA, coding for a protein that is 1,863 amino acids. I see there are very many variations of that particular transcript. You might also notice that the gene has an identifier that begins with ENSG. And ENS stands for Ensembl, and G stands for gene. So ENST stands for Ensembl transcript. ENSP stands for Ensembl protein. Now, if this were a non-human gene, there would also be a species identifier, so it would have a longer. For instance, a mouse gene would be ENSMUSG for Ensembl mouse gene. But human, by default, gets fewer letters. If you scroll down, we'll see all of the different transcripts. And you might also notice a little caret. And a little caret at the prior to BRCA1-001. And what that tells me is that this gene is read right to left, not left to right. So it's on the complement strand. And you see some boxes that are filled. You see some boxes that are empty. All of the boxes represent exons, and all of the connecting lines represent introns. The empty boxes refer to the untranslated regions, and the filled boxes refer to the coding regions. There are different colors that are based on the different evidence for the specific genes. There are also the blue gene models are usually non-coding, and the red and the gold are protein coding transcripts. Okay, we can also see the location. It's on chromosome 17. From position, this means nucleotide 41,196,312 through 41,277,500. Again, it says reverse strand. I know it's on the reverse strand, again, because the caret points to the left, so it's a right to left gene. Also, there is a blue bar that represents the contig. The contig is the piece of DNA that this gene is on. And Usually when the gene models, or always when the gene models are below the contig, it's a reverse strand gene. If the gene models are above the contig, like you see with this NBR2 gene, that is a plus strand gene, and that reads left to right. So I'm going to click on that NBR gene, and I'm going to open that gene record. There's the ENSG. So here's the NBR2 gene, and, well, it's really creatively named. NBR2 stands for neighbor of BRCA1 gene. It's a non-protein coding gene, so this is an RNA product that doesn't produce a protein, so its function is probably somewhat unknown. Actually, it said it's a known pseudogene, so this is a remnant of a gene that used to exist. In this particular gene, there are 4 different transcripts, none of which are coding. And we see now that I'm centered on the NBR2 gene that it's above the blue bar and the caret points to the right. Now let me go back to BRCA1 and click the ENSG link again. Okay. So I can either show the transcript table or hide it. If I hide it, it disappears. If I show it, you see the table with all of the known transcripts. So I'm going to click that first transcript ID to show you what the transcript page looks like. The gene page is focused on the entire gene. The transcript page focuses on one particular mRNA. And if I look at the left menu, I can find out a few things about it. For instance, there are 23 exons in this particular transcript. And if I scroll down, I can see those exons here. Remember, we're reading right to left here, so the first exon is on the far right. The last exon, which appears to include the stop codon, is on the far left. I know that the stop codon is in that last exon because part of the box is filled in and wider. The rest of the box is empty. So after the stop codon is an untranslated region that's not part of the protein product. That's the empty box. There are 23 exons. 22 of them are coding. The first exon is completely within the 5' untranslated region. So the start codon would be in exon 2, which would be the second box from the right. Okay, now if I want to know the sequence of those exons, I could click exons on the left menu. And that shows me ENSE. Remember, ENST was Ensembl transcript. ENSE is an Ensembl exon identifier. And here is the exon sequence of exon 1. Remember, this would be starting from the right. And purple indicates untranslated. And the purple ends in the second exon. And the first 3 nucleotides in black are ATG. And ATG in RNA would be AUG, so AUG is a start codon. That represents the beginning of the coding region. Now, the intron sequences are usually truncated unless you manually turn them on, because typically you're more interested in the exon sequence than the intron sequence, so you see that there's sort of a dot, dot, dot. Usually intron sequences tend to be much longer than the exon sequences. But this allows you to quickly see the exon sequences. The exons, the letters are capitalized. The introns, the letters are lowercase. And you can see it looks like exon 10 is very, very large. And then in the final exon, you see the TGA, which is a UGA or a stop codon. After the stop codon, it turns purple. That's the 3' untranslated region. So you can get the sequence by clicking exon. I can also click variation table. And variation table will take me to a table which shows every known variation in this particular gene, meaning that maybe some people have a different nucleotide at this particular position than is shown in Ensembl. And the variation tables show different types of single nucleotide polymorphisms, or SNPs. You'll see that you can click show to show all of them. A 5' untranslated region variant means that there is a variation in the population that affects a particular position that is before the start codon. Now, it looks like there are 947 known variants that change the coding sequence. So if I click show, it will show all of those 947. And believe me when I say that the explosion in sequencing technology has increased the known number of variations almost exponentially in the last 2 years. This table used to be a lot smaller only a year ago. And here are many known amino acid variations. And you see that 1858 means at amino acid 1858, there is a coding sequence variant. Now, if I'm interested in the protein that is coded for by this particular transcript, I could click Protein Summary on the left menu. Okay, and the protein summary, I'm going to now hide the transcript table so I can see it, shows what particular domains are present in this particular protein. So the superfamily domain has noticed a particular type of region here. It looks like there is a zinc finger domain at the N-terminus, which is at the left. Proteins typically are described from the N-terminus on the left to the C-terminus on the right. There's a BRCT domain, which is involved in breast cancer at the C-terminus. And you see that the sequence variants are shown. These are all the SNPs, and there are tons of them. You also get some information about the protein itself. Its charge is negative 45.5. Its isoelectric point is 5.1, so it's an acidic protein. Its molecular weight is 207,720, and there are 1,863 amino acids. Okay, now if I want information about the domains that are shown in this table, you see protein domains for the ENSP. I could click Domains and Features, and that would give me links to some of these databases. So you'll notice that there was a superfamily match from position 1 to position 103 in the protein. And there's an SSF accession number. When I click that, it says that this is a ring finger domain, which is a common fold that's found in some proteins. The external documentation suggests that there's a ring finger domain from position 1 to position 103. You see a lot of information about the BRCT domain from many different databases, both Superfamily, ProCite, SMART, PFAM, and there are individual links to those external databases to documentation about that particular domain. So I'm going to click the PFAM domain for the BRCT PF link. And BRCT domain is named after the C-terminal domain of a breast cancer susceptibility protein. So BR for breast cancer and CT for C-terminal. And it's found predominantly in proteins involved in cell cycle checkpoint functions. And you see that it's involved in proteins that are responsive to DNA damage. That's not surprising that a cancer protein might be related to DNA damage. And you get some information about the structure of the protein itself and some other proteins that actually contain that domain as well. So the BRCA1 protein is not the only protein that has the BRCT domain. Here are a list of others. Now, if you notice, there are a bunch of tabs at the top. One says human, one says location, one says the gene, BRCA1, one says transcript. I'm going to go back to the gene record. To do that, I'll just click gene, the tab, which is the third tab from the left. And let's say that I want to look at related proteins in other species. On the left menu, it says orthologs. It says there are 60 orthologs, which means that there are 60 known relatives of the BRCA1 gene in other species. So I'm going to click orthologs. And you can see an ortholog summary, some from primates, some from rodents, some from fish. And here are a list alphabetically of all of the genes that are related to the BRCA1 gene. You see alpaca, lizard, armadillo, cat, chicken, chimp. You see really just about every species you can imagine. Now let's say I was interested in the mouse equivalent of the BRCA1 gene. So here's mouse mus musculus. And as I mentioned, it's instead of ENSG for a mouse gene, it's ENSMUS for mus musculus is where the MUS comes from. And there's a different identifier for its gene. So I'll click the mouse BRCA1 gene. And what you notice is that rather than over 30 transcripts, instead of 31, there are only 4 found in mice. There were only 4 or so in human that were known about 2 years ago, but because there's been so much attention in recent years devoted to the BRCA1 gene, they have found so many more transcripts. There probably are more in mice as well. So what you notice is that there are 4 transcripts. 3 of them do not code for a protein. 1 of them does. It's an RNA that's 6,572 base pairs and 812 amino acids for the protein. It is also on the reverse strand. It points to the left, so it's reading right to left in mouse. And you see that it also has one very long exon in the middle. The gene pattern looks relatively similar. So if I click splice variants, we'll be able to look at the variation in mice. And you sort of see a blow up of the exons. The yellow gene model is the protein coding one. The blue gene models have all empty boxes because they don't code for protein. They're just processed transcripts. Why they exist, in some cases we don't know. In some cases we do. But it's not uncommon for genes to have unprocessed transcripts, splice variants that don't code for anything. Well, here's the BRCA1 gene, and you can see, remember we're going right to left, the first exon is completely non-coding, and then you see that the stop codon is in the final exon on the left. You also see a quick look at some of the domains that are present in this particular gene. And again, if I click the transcript, just like in human, I will get the entire gene model a little bit blown up and a little bit more information about that particular transcript with links on the left menu. This concludes our tour of the Ensembl database. Like any database, the best way to become familiar is to find some genes, look for them in a particular organism, and explore. Good luck.